My pleasure to introduce two speakers this afternoon from the University of Glasgow, Rachel Jack and Philippe SCHYNS, who are here for a visit as part of their Cross America tour. They were in San Diego yesterday, and flew to New York and then drove up here in a great big American car with a four-wheel drive. So Rachel Jack got her PhD at the University of Glasgow in 2007 with a thesis entitled "Cultural Differences in the Decoding Representation of Facial Expressions and Emotions." And she will be the lead-off speaker. And then she'll be followed by Philippe SCHYNS. And Philippe got his PhD at Brown University back in 1992. And then proceeded from there to be an assistant professor at the University of Montreal and then at Glasgow. And you were at MIT someplace in the too, weren't you? Okay. So without further ado, I'm going to give them plenty of time, because I know that they don't want to encroach on each other's time, and I don't want to encroach on theirs.
So yes, thanks very much, Jim, for the introduction, and good afternoon, everyone. Thanks very much for coming. Thanks, Jim, also for organising our stay here. It's a real pleasure to be here. So as mentioned, our talk's going to take two parts. The first part, we're going to show how we use psychophysical methods to model dynamic facial expressions of emotion. And in the second part, we'll show how similar methods can be used to examine information processing in the brain. So I just first want to thank the people who fund me or fund us, or have funded us. And also, we have really great collaborators in Glasgow, but also in other institutions. I think I should also be adding Talia down here somewhere, because we have a text paper to write. Okay, so the face is one of the most powerful tools in social communication, and it has a number of independent muscles that can be activated different intensities across time to create a variety of different complex dynamic patterns, and these are otherwise known as facial expressions of emotion. And much like language, facial expressions, so their origins, their meaning, their syntax, have been studied for over a century in fields like psychology and philosophy, and much more recently in computer vision and, say, social robotics. And most notably, based on Darwin's groundbreaking theory of the biological origins and evolution of facial expressions, a wave of empirical research pioneered by Ekman reported that 6 specific facial expressions of emotion are recognized across all cultures. So you may recognize these 6 what's called FACS-coded or standardized facial expressions of emotion. And these were stimuli that then became the gold standard in emotion research. However, what is also the case since Ekman's 1969 science paper is that these particular facial expressions have not been recognized with comparable accuracy across all cultures. And in fact, what you find is that while Westerners tend to recognize them with very high accuracy, in other cultures they have significantly lower recognition performance. So this map here just shows with the color coding here, the accuracy using these 6 facial expressions of emotion, and those that are below 75% accuracy, which would be considered roughly kind of typical performance, are outlined in black. And you can see here that there's quite a lot of variability in recognition performance with these stimuli. So this brought me to think that if these particular facial expressions are not well recognized in other cultures, they don't represent these different emotions, then what do these facial expressions look like? Okay, what do they look like in other cultures, and how do they communicate these concepts? So we have addressed this question using a psychophysical twist on the classic method of using the observer to inform knowledge about facial expressions. So let me just illustrate the method that we use. So imagine that you encounter these 2 facial expressions. You might think that the person on the right-hand side is disgusted, whereas the person on the left-hand side, you might think well, I'm not really sure what they feel there. Doesn't look like anything I know. And that's because the dynamic pattern that's presented on the right corresponds with what you already know about a discussed facial expression, whereas the one on the left doesn't really correspond to anything that you know. Well, some people say like skeptical or something like that. So by showing animations like these to observers and asking them to categorize them by, for example, the emotion categories, we can identify the dynamic patterns that communicate those concepts to an individual observer in a given culture. Now, of course, the trick here is that both of these animations are actually randomly generated, and that's really the basis of our method. We randomly generate facial movements and allow the observer, not the experimenter, to choose the facial expressions that communicate different social concepts in their culture. So let me show you now how we generate these facial expressions. So to do this, we built a facial expression generator. So this is, I say we, I had nothing to do with it. This is Oliver, who's a co-creator of the GFG. And we recruited a group of fax coders. And fax coders are people that are specially trained to move individual movements or action units on their face. And we had them move each of 40 core action units here, represented here, and we recorded them with a stereoptic system. So then we have a model of each action unit from these fax coders. We then parameterized each individual action unit to control their movement. So then by combining action units that have a dynamic pattern together, we can create almost any facial behavior. So for example, on each experimental trial, the platform will select a random set of action units, anywhere between 1 and say 6 action units. Here we have nose wrinkler, we have eyelid raiser, and then this is lip corner puller. And then for each action unit individually, the platform will select 6 random values for the temporal parameters, okay? So you have, say, onset latency, acceleration, peak amplitude, peak latency, deceleration, offset latency. So these are random values for each of the action units separately. The platform then combines these randomly activated action units to show random facial movement. The observer, who is then naive to the random nature of the facial expression, will then categorize it according to, for example, the 6 facial expressions of emotion. If it doesn't correspond to one of these labels, or if it's a combination of these labels, they're asked to choose other. They only respond if this matches one of these labels. So then following the experiment, what we have for each individual observer is a set of animations that they have categorized as one of the 6 emotions. And then we can identify the action units that are significantly correlated with that perception. And then for each significant action unit, we then identify the specific temporal parameters using, say, a linear regression. So we use this platform and this approach to model dynamic facial expressions of the 6 classic emotions in 2 cultures in a 2012 PNAS paper. We had 15 Western Caucasian observers, 15 East Asian observers, and they saw around about, or they categorized around about 5,000 of these facial animations presented on same and other race faces. So I'm just now going to show you a brief illustration of some of the results. So this video is going to play on a loop, so we have, I'll just catch it, happy, disgust, and anger. And let's start with the East Asian one. So here we have East Asian models of facial expressions, and we have the Western models. And you can see that the East Asian eyes vary a lot between the different emotions. So we have happy, disgust, and anger. So you can tell the difference between all 3 expressions very, very easily just on the eyes. You can see for anger we have the eye whites. See? Should we go again? Let's go again. Happy, disgust, anger. That's really easy. Let's look at the Westerners now. So we've got happy, disgust, and anger. These are virtually identical. You just can't tell the difference between, say, disgust and anger with the eyes only. So let's now switch to the mouth. And let's start with the Westerners this time. happy, disgust, and anger, okay? Happy, disgust, and anger. So there's some variance in there. You can tell the difference between disgust and anger just from the mouth with the Western models. Now let's look at the East Asian group. Happy, disgust, anger. It's virtually identical between disgust and anger. You even have this asymmetry in the lip corner puller, in the lip raiser. So here we show that Easterners and Westerners use different parts of the face to communicate different emotion categories. I just want to have a really quick note here to say that we've used this platform to look at cultural differences in facial expressions of emotion, but we're also use it to look at commonalities as well. And so we've recently modeled over 60 different emotions across 2 cultures, and we find that there are actually 4, not 6, culturally common action unit patterns. That's just for another day though, I think. And we've also recently extended our work to look at facial expressions of different mental states. So we have emotions, but we also have mental states like interested, bored, and confused. And we found that both interested and bored are actually very culturally similar. They use very similar action unit patterns to communicate these different social concepts in Western and Eastern culture. Here in interested, what we find is that we find facial expansion movements, whereas we find in bored more contraction movements, sort of closing of the eyes and so on. In contrast with confused, we found that while Westerners used more of the lip stretcher, East Asians used this jaw drop here. And when in fact, when we found these results, my PhD student, who's a lead author here, we saw this. She says, I don't make that face. Says, yeah, you made that face all the time, and now I know for sure what it means. She was not a subject here either. We also then show that differences or convergence and divergence in these action unit patterns significantly impacts cross-cultural communication. So here what we have for interested and bored, the red areas show where cultures converge, and the blue areas show where cultures diverge. They show the action units. So interested in both, interested and bored, are very culturally similar, and we can see that we have high cross-cultural communication accuracy. In contrast with confused, we have dissimilarity, and then what you have here is impacts negatively on cross-cultural communication accuracy. So aside from the cultural aspects of my work, we've also used the platform to examine how and possibly why facial expressions transmit information over time, because they're a dynamic stimulus. Why should certain action units, for example, come first and others come later? Why should they have evolved in that way? And so in a 2014 current biology paper, we used a combination of information theory and Bayesian classifiers applied to just over 700 dynamic facial expression models. And what we found is that facial expressions of emotion transmit information in an evolving hierarchy across time. So let me just show you a couple of videos to illustrate the point. So for example, in fear and surprise, early in the signaling dynamics, they both transmit the eye whites, but only later do they transmit face movements that distinguish them. So we have brow raiser and mouth stretch that actually distinguish them later in the signaling dynamics. So again, we have a hierarchy across time of shared and distinct action units. We also found a very similar pattern with disgust and anger. So early in the signaling dynamics, they both transmit the nose wrinkler, and they're only later distinguished based on upper lip raiser and the eye whites. So I've shown you some of the work that we've used with the dynamic aspects of the platform, but we've now recently extended this to be able to sample information from other aspects of the face, for example, like morphology and complexion. So as you can see, this is my face, and we are transforming my face in accordance with different genders and ethnicities and age. We also use texture or complexion, where we can sample information and then transform individual faces in accordance with different ethnicities, age, and gender. So we can use this platform to sample information as we do with action units to then model make models of social face perception that integrate dynamics, morphology, and complexion. And we can use this then to address key questions in social psychology, such as the in-group advantage, racial stereotyping, and say the other race effect. And finally, we aim to then transfer the models that we have, our models of social face perception, to social robotics in order to design socially sensitive and culturally aware digital avatars. And for this, we're hiring. So if you're interested, come join us in Glasgow for some fun. Okay, thank you.
Okay, thank you, Rachel. So we'll now pick up the talk where Rachel left it with a framework, which is the framework that we use in Glasgow, whereby our first aim is to discover what information behavior requires in order to make a categorization. So what you have seen today is the latest expansions of our reverse correlation platform. We originally started with pixels, and as I will show you now, because our aim is from having a lower bound on the information that's used for behavior, we try to dig in the brain to understand where, when, and how the specific information that's used for categorization is processed. So our aim is first to reconstruct a network where the information is computed, and then try to reverse engineer the mechanisms whereby visual coding, memory, and decision happen. Of course, that's a tall order. That's the big, broad picture. And what you should see from now on is maybe demonstrations rather than very firm results about visual coding, memory, and decision. So as I said, whenever we try to develop the platform, in this case for the brain, we go back to old school bubbles. Now, old school bubbles is not about sampling facial movements, but rather about sampling pixels. So going back to the original experiment to fix IDs, we showed stimuli of that sort, which are simply pixels of females and males, each being neutral or smiling. And we ask in one task observers, do you see a male versus a female? Here you would probably see a male, here a female, and here you might find it difficult. Don't mind the haircut, it's been normalized. Then we would change the task and ask the same observer, is the stimulus happy? Yes. Or neutral? Yes. And here they probably wouldn't know. Then, following the experiment, we basically build a relationship between the pixels that we sampled and the response of the observer, with the idea that when they said yes, we showed the pixel revealing the diagnostic features, such as the mouth inexpressive or not, or the eyes in gender. Of course, we can discover these pixels because sometimes we don't know what the features are for a given task. We can, of course, do the same analysis with a continuous measure rather than a discrete response yes/no, in this case reaction time, simply by, for example, dichotomizing the reaction time distribution from the median, and again build the relationship between the pixels that led to fast responses, such as the mouth, and those that led to slow responses, which would have been the absence of the mouth. And likewise here for gender. Okay? So now, how do we use the same methods to look at the brain? Well, essentially, it's very simple. It's, again, a problem of building a relationship between the pixels that are sampled and a brain response. You can build this relationship within a response. A voxel, we've done that. Or you can move to dynamic responses and look at EEG sensors, which you see here, I will explain, or MEG sources, which you will see later. So how do we do this? Well, let me start, say, from time point 204 milliseconds following stimulus onset. This particular electrode, P10 here, is one occipitotemporal electrode. It measures the temporal lobe quite well, the activity that's going on there. P9 is just the opposite electrode. It could also be called P8 and P7, depending on which electrocap one uses. And what I'm illustrating here, on this column here, precisely at 204 milliseconds, is the relationship between the EEG voltage on this axis and the information that was sampled on each trial. So specifically, if a given sample of pixels gave rise to a very negative voltage on that electrode, I would add the samples here. If, on the other hand, another sample of pixel gave rise to a very positive voltage, I would add the samples here. And in this manner, I progressively build a distribution of how the pixel sample co-vary with voltage. And what you can see here is that as we go from mean voltage down to the most negative voltage, we see the appearance of the left eye of a face. That's the eye that's contralateral to the measuring electrode. Yes? As we go to the more positive voltage, which are the worst voltage for this particular response, which is the N170, we see the disappearance of the eye. So the eye wasn't present, always contralateral to the measuring electrode. We move to the other electrode, and we see just the opposite pattern. As we go down in voltage, we see the appearance of, this time, the right eye, which is contralateral to the left measuring electrode. Yes? So once we have established this mapping at 204 milliseconds, we can formalize it. How do we formalize it? For example, by building a little linear regression between each pixel and the voltage response, or by correlating it, or even more simply, by summing all the pixels leading to a positive voltage, summing all the pixels leading to a negative voltage, and building a difference score, with a null hypothesis being that there will be no difference. Right? So, or we can even use mutual information, as I will show later, which is an information theoretic construct. So basically what we do is we build one of these measures of association and formalize it and represent it here to substitute for the average. So we can then show which information at a given time point, 200 milliseconds, modulates the EEG response, the contralateral eye to the measuring electrode. Then, because we have a dynamic response, we repeat this with the resolution of our response, which is here about 16 milliseconds. And then if we follow the mean, where we look at what's called the classification images, we can see the progressive appearance of the contralateral eye response. So we can conclude from this that in the gender task, for this particular observer, we always do this per observer, the right electrode reflects a response to the contralateral eye of the stimulus. So the EEG codes this response. Likewise, for the other electrode, the left electrode codes the right eye. And you'll say, but that's good, because we had in gender, if you remember, as diagnostic information, usage of two eyes. So are we seeing here the time point at which the brain codes the information that's diagnostic for gender? If this were the case, the same subject categorizing the same faces according to their expression, where the mouth mattered, should code the contralateral corner of the mouth. And no, that's not what happens. Rather, we still see a contralateral coding of the eye, maybe some little evidence of the mouth here. But if we switch electrode, going back to expressive or not, and use this time parietal electrode PZ, and change time, this time we are starting from about 214 milliseconds, leading to 400. We see that as the P300, which is another major event-related potential, unfolds, the information that leads to the best, the highest, most positive voltage values are this time correlated with behavior. So you see the mouth in green associated with high voltages, and for the same subject, you see the two eyes in green associated with high voltages. So what can we conclude from this old school bubble study? Something quite simple. If we use behavior together with bubbles and build the association, we find a task modulation of the information that's required for behavior. We do not find a reflection of this task modulation in the information the brain codes early on along the N170 ERP, but we see a task modulation of the information that the brain codes on the P300. Okay, that's clear for everyone? That's really old school bubbles. Since then, of course, we've done more than this, and we've, for example, delved into facial expressions of emotion to show that the N170 in fact reflects a much more complex process. Our task was very simple. This is a more complicated task of 6 choice of the 6 basic facial expression of emotion that Rachel showed before. And the mechanism that we were able to demonstrate alongside the N170 was indeed a mechanism of task dependence, unlike the first conclusion we had before, whereby we find coding of features contralateral to the measuring electrode that tend to start in the eye and then move down on the face until the feature that's diagnostic of the expression has been coded. So in fear, where you typically have 2 big eye whites, you will see coding of the contralateral eye, and then the mechanism will stop there and we will have an early ERP. In disgust, such as is here, coding will start in the eye and then move down on the corner of the nose, and you will have a delayed ERP. And in happy, which typically involves a smiling mouth, coding will start in the eye, move down on the face until the corner of the mouth is found, and you will have an even more delayed ERP. So the distance between the eye and the diagnostic feature of an expression determines the latency of the N170. So we gently go into coding mechanism here, which are quite detailed, I hope. And you can notice that in both cases, we still preserve contralaterality. That is, you see here in blue the coding of what the right electrode does. It codes the left side of the face primarily, including the corner of the nose towards the end of the N170. So we are here. Whereas the left electrode codes the right side of the face primarily. And that goes, of course, with the early architecture of vision, which is split by hemisphere for contralateralized hemifields. So we have some kind of sanity check there, that we're not just looking at something that's random, but something that fits what we know about vision. Now, this is the first time I present everything that unfolds, but hopefully, with what you have seen so far, we can move quite quickly. Now, what we want to do, from this starting point of knowing that different electrodes or different sources demonstrate coding of features that are or not task relevant, we want to add one element to this. Can we find methods whereby we can trace the flow of diagnostic information in the brain? The answer is yes, and we'll proceed in 2 steps. The first step is a paper that has just been submitted, is one where we had observers performing a face detection task using old school bubbles. It's always useful to go back to basics when we want to develop new methodologies, because we have a lot of information that guides our hypothesis. What you see here is the left occipital electrode, I think P7 here, the right occipital electrode P8, sorry. What you see here is the N170 on this electrode, here the N170 on that electrode, and reflected in red and blue, respectively, the coding of the contralateral eye, as I have shown before. We are just switching the representation to have something that's easier to see. Basically here, the magnitude of the coding in question is directly reflected by the intensity of the color. Right? So the first peak for coding is at T1 here for the contralateral eye, and in parallel you have coding of the other eye here at T1. Yeah? But if you look very carefully, you will notice on the right electrode and also sometimes on the left electrode that this electrode also codes another feature, namely in this case the ipsilateral eye to the right electrode, so the right eye, at T2. So there's a gap between the coding of these two features. So this immediately raises a very simple question of, is the feature coded here at T1 on the left electrode transferred across hemisphere to the right electrode where it appears coded at T2? So basically, do we have a causal transfer of a feature following its coding? There are three conditions to satisfy to determine that we have causal transfer. The first is, well, obviously, we have to have coding of the same feature on two different electrodes or two different MEG sources, as I'll show later. We have to have temporal precedence of this coding, so it has to happen here at T1 and then later at T2. And then, and that's the most crucial thing, the coding must be redundant between the two electrodes. What do I mean by this? I mean simply that if we modulate the visibility of the eye, the two electrodes, the responses on the two electrodes or the responses on the two sources should be modulated in a similar manner. So let's go through the conditions. Feature coding, it's quite easy to convince you, I think, because I've shown you the fully developed coding before. But one simple way to do it is to compute on each single trial where we sample with bubbles the visibility of a given feature. So here the visibility of the left eye, and here the visibility of the right eye. We represent in white the trials on which there's very low visibility of the feature of interest, and in a more saturated color, the trials on which we have the highest visibility of the feature. And then we plot the ERP for each one of these bins of trials. And as you can see, the ERP shape changes considerably as a function of the visibility of the feature. So we find feature visibility in the ERP, as coded by the ERP. We can translate this into a measure of relationship, as discussed from the beginning. That's what interests us, which could be a correlation, if we wanted to, between feature visibility and EEG amplitude at each time point. But here we chose to do this with mutual information, which measures in bit the relationship between two variables. And you can see here in bits that there's a strong relationship between the visibility of the eye and the EEG, as expected. And likewise here, on the same electrode, we have the strong visibility of the ipsilateral eye, and it peaks a bit later. And then we represent this as you have seen so far. We overlay with a saturated color, representing the mutual information, the relationship between visibility of the eye and the EEG. And we have a first peak at about 140 milliseconds to the contralateral eye on the right electrode, and then a second peak at about 200 milliseconds to the ipsilateral eye. And of course, we would have something very similar on the left electrode. So we do find coding of the same feature across two electrodes, and that's good. But we also find a systematic latency in this coding. Each dot here represents an individual subject. When you consider the latency as represented here between, on the right electrode, between the ipsilateral and the contralateral eye, and on the left electrode between the ipsilateral and the contralateral eye. Basically, this shows, in a nutshell, that we have about 15 milliseconds of delay in the coding of the same feature. between the two electrode sites. And this was true in 13 out of 15 observers. And this is commensurate with typical timing of interhemispheric transfer. And then the last part is the redundancy. Now, redundancy is formally expressed as the intersection of the mutual information between the EEG at T1 on the left electrode and the right eye, and the mutual information of the right eye, again, it's the same feature, at T2 on the right electrode. So basically what we want to show here is that this coding in the EEG and that coding in the EEG of the same feature has a high redundancy. Simply put, with Venn diagrams, the way we do this is we add the two mutual information measures as if they were independent. So if they have redundancy, as expressed by this little darker red here, they will add together. And we subtract to this the mutual information of the joint variable, left and right electrode, at T1 and T2, and the right eye, where we count the redundancy only once. So this would be 1 plus 1 minus 1 equals 1, if it was perfect redundancy. And that would be 100% redundancy. If there was no redundancy, we would just have nothing. Okay, so now what we are seeing here is the evolution of redundancy, the redundancy measure, using a first time point here on the left electrode as reference, and we compute from this time point, which is the maximum of the coding of the right eye, on the left electrode, we compute redundancy with all other electrodes to see where and when redundancy evolves. And as you can see, as time develops, we see the first sign of redundancy after about, as we know, 15 milliseconds on the other electrode, the right electrode, if we start from the left. And if we start from the right, we see on the left electrode, whoops, the redundancy appearing to the ipsilateral eye. Yes? And this measure of redundancy that can be expressed as a percentage across all 13 observers who satisfied the first two conditions, as you can see, is high. It's from 40% up to 90% redundancy. So basically, there were three conditions for causal feature transfer between two electrodes. One was coding of the same feature. We've shown this, and you see it illustrated here, with coding of the right eye on the left sensor and on the right sensor. Temporal precedence of the coding, indeed, the peak here precedes the peak there for the coding of the same feature. And then redundancy of the coding, which I've shown before, exists for 13 out of 15 subjects. So we do have, in a large majority of observers, evidence of feature transfer. So yes, indeed, we can start to follow the flow of information in the brain that is important for a task. Here, the eyes are important for face detection. If you are still with me, now we want to develop this one step further. We want to develop a method of directed feature transfer that will enable us to do the same sort of thing, but for any possible lag between any possible sources. But you will see that we will still go back to the same three conditions of coding of the same feature on two sources, temporal precedence between the two sources, and redundancy. The redundancy will be expressed slightly differently here. Okay, so again, we go back to old school bubbles, an old favorite, which is a painting of Dali, where we will extract this little part. We will sample the stimulus, splitting it into 5 spatial frequency bands of 1 octave each. We will apply bubbles to each one of the spatial frequency band to reconstruct a stimulus like this, where the observer will be able to say, I see Voltaire, which they would probably do here, the bust of Voltaire, or I see the nuns. For those who do not know, these are the two faces of the nuns with their dresses. Can you all see the nuns? Yeah? Or I just don't know what I'm seeing. It's neither of them. Then, following our approach, we want to know what information drives these perception using behavioral measurements. So here, using mutual information, for each of the sampled pixels, we will build the mutual information between pixel sampled and the nuns versus not knowing what's in the stimulus. And when you do this, you see that indeed the two faces of the nuns come up across two spatial frequency bands. For Voltaire, we have a slightly different profile of mutual information. It's primarily what you see here, which is the low spatial frequencies that represent the bust of Voltaire particularly well. So when we are here, we know that the brain must use at least this information to perceive the nuns of Voltaire. So we have a lower bound on the information that we will look for in our networks to see how these are transferred, processed and transferred. For sanity, what we do is an independent analysis on all the brain sources. So while the subject did the experiment behaviorally, we measured their MEG. From the MEG activity, we reconstructed sources with ICA. And then what we have is a massive matrix. On the y-axis, we have all the sources that we have found. On the x-axis, we have time. And for each cell, we build a mutual information image between the pixel sampled and the cell response. Having this massive matrix, we perform a dimensionality reduction using nonlinear matrix factorization, and we find that the main features that the brain process in the task are the left face of the nuns in blue, the right face of the nuns in red, the main Voltaire feature in green, and interestingly, we find also that the brain processes other features than those that are strictly necessary for the task. We can also see these. Okay, then what we want to show is what? Well, we want to show that source A codes, for example, the left nun face, and sends this feature to source B. So let's go back to our reasoning. First, we must show that A codes the same feature as B with a temporal lag. Something like this. So we have here all the MEG trials of one source, which is A. We have here all the MEG trials of source B. And in red, you see the activation of the source, obviously. By the way, that's an example. It's not a real one. And this is to illustrate the mutual information measure that we build between the visibility of the trial, of the left nun's face in the trial, and the corresponding response of the source. When we compute this mutual information at each time point, we find a peak at T1 here, where this peak means that there's a strong relationship between the visibility of the left nuns and the MEG response. And here, there's no such strong relationship. On source 2, we build the same mutual information relationship, and we find at T2 a strong relationship. So what we have is then coding of the same feature, the left face, with temporal precedence between the two sources. The first two conditions are satisfied. They are the simplest ones. And now we go to the other one, redundancy. Okay. So if you remember, our aim was just not to look at peak-to-peak relationships in redundancy, but rather be able to examine all of the lags. The lags are represented here. And we go from, say, 30 to 200 milliseconds between two sources. This is time. I've represented here this T1 and here this T2. And what you see here is what's called directed information, which is also called transfer entropy, which is also called the conditional information, the conditional mutual information between the responses of two sources that's conditioned on the path of the receiving source, so that we can show that there's a transfer of information across sources that's independent of the path of the source. That's basically transfer entropy. And we can just see it very simply like so. So it basically means that if this source A increases in response, source B increases in response, so there's a relationship, but you have to remember that it's conditioned on the path of the receiving B source at the same time point T1. So what is this telling us, directed information or transfer entropy? It's just telling us that two sources communicate. And here they communicate with one source sending information at T1, A, and B receives it at T2. That's the little blob that you see here. So there's a lag here of about 100 and some milliseconds. Okay? So that's all the communication that takes place between two sources, including possibly communication of a feature. So how do we isolate, and that's the critical part, the communication of the feature? Well, we simply, quote, quote, repeat the same computation that you see here, but we withdraw from it the computation of transfer entropy that is conditioned on the visibility of the feature. By conditioning transfer entropy on the visibility of the feature, you are doing basically the same as when you are computing conditional probabilities. You remove the effect on transfer of the feature itself. Okay? So you have all transfer left except that of the feature in this second part here. And now this is where information theory can be very counterintuitive. Here you have all the transfer between the two sources, including the feature. Here you have all the transfer excluding the feature. So by subtracting one from the other, what is left in blue here is the transfer that can be attributed to the feature. So the feature transfer. Okay? If you've not followed any of this, it doesn't matter. What matters is this. By now, we have a measure between two sources where we can say with reasonable confidence we can measure the transfer between two sources of a given feature at a given lag in time. So what can we do with this? Well, remember, we had many sources. I'm representing here only a few to start. This source is, okay, so this is the back of the brain, left hemisphere, right hemisphere, and we are seeing it from the top. This source is roughly occipital. This one would be maybe near your FFA. It's difficult to tell, it's MEG. And this one is also occipitotemporal somewhere. What you see here is time. This is mutual information. And the three colors represent, together with a pie chart, the features that the source codes. So this source codes, for example, at 124 milliseconds, it peaks by coding the left face of the nons. But you can see that this other source codes the same feature with a peak at 188 milliseconds. And the transfer of feature measure reveals that there's a communication of a feature between these two sources. So we can conclude this source sends the left eye to that one where it arrives at 188 milliseconds. The same source at 140 milliseconds codes the right nonce face and sends it to the same source here where it arrives at 203 milliseconds. So this source now starts to act like an information hub where we see convergence of information possibly from some region of primary visual cortex, though of course then you would like to see independent sources in primary visual cortex, I agree, but this is MEG. And then we see later on this other source here sending at 164 milliseconds the Voltaire feature, where it arrives at 196 milliseconds. So indeed, this source looks to appear like a hub. A lot of information converges on it. Can we see information diverging from it thereafter to converge elsewhere? Yes, we do. So source C here codes the right nonce phase and the left nonce phase, but quite late. It's a lower parietal source, and it should be reminiscent to what you saw earlier on electrode PZ, where we could see task coding. And what you see here is from 180 milliseconds on this source, and from 188 milliseconds on this other source, a transfer of features to the parietal source, where they arrive at 300 and 316 milliseconds. Okay, so what have we shown here? Well, basically not much more than some kind of existence proof that we could reconstruct sources, show that they code features that are important for perception, and then start to trace the flow of information in terms of feature transfer in the brain itself. So just to summarize what we have tried to show you today, we start with the first question of what information does categorization require? And then ideally what we do is that we build the ideal sampling platform to the task that interests us, so as to be able to characterize very precisely the information that's important for behavior. And you've seen quite a bit of what we do, ranging from very simple bubbles up to, as Rachel showed, sampling of facial action units in motion. And what you've also seen a bit is sampling of multivariate identity components, though we haven't gone in there much, to represent identity. So when we characterize this, we derive a lower bound on the information the brain must process in order to categorize. And then we try to reconstruct an information processing network. We have used primarily EEG and MEG, but we'll move into fMRI for better localization, obviously. We've burned our fingers quite badly so far with MEG, but we are improving there as well. So then we have a network of sources, and we try to understand which information these sources code, where they send their information, and then when and how all of this happen in the context, because of course we haven't seen any mechanism yet that are very precise, of visual coding, memory, and categorization decisions. Thank you very much. Just on time.
We have time for questions.
In one of your last slides, if I understood correctly, you showed that the faces of the nuns get processed before like Voltaire's face, right? So is it the case that the high spatial information gets processed before low spatial information?
It's a very good question. I'm one of those responsible for the course to find strategy of recognition with the scene hybrids that I did back in 1994, where we showed that very brief presentation of a scene hybrid with Odoliva was recognized on the basis of the low versus a longer presentation on the basis of the high. And then ensued a number of beliefs, because the second paper that we published in '97 showed that this was not the case. We could very easily flip via attentional manipulation to the high or low spatial frequency bands. We have run a good number of bubble studies, actually, that looked at this, and I can send you a few papers, but what we find quite a lot, but remember that observers are aware of the task they are doing, yes, always. There's nothing by surprise here. So in the context of facial expressions of emotion, you tend to find along the N170 a time course of scale processing which starts with the eye, contralateral to the measuring electrode, in high spatial frequencies. Then when people move down on the face, if there is one such thing, you can see that we are moving full spectrum. And then when they go back to focus on the diagnostic features, they come back to a high spatial frequency representation in the classification images. So I think the picture is a lot more complex than many people would think. But of course now, if people did not know what the task was, and they were to see something, well, they would go for what's most informative, which could be, at least for the task in question, given the constraints of vision, low frequency.
I had a question for Rachel, actually. I was hoping you could elaborate a bit on your conclusions that you drew from the expression experiments involving cultural differences. I was struck by the fact you pointed out that we could easily read the Asian faces, the differences there when reading the eyes. So I just wanted to understand better what your conclusion was.
Well, with this particular result that I showed here, it was really to point out that different cultures use different parts of the face to transmit emotions, right? So Asians, the eye region has more variance in signals, whereas with Westerners it tends to be more variance in the mouth. Of course, both cultures use the eyes and the mouth, but the variance tends to be located in different parts of the face.
And just to add to this, it's reflected in the emoticons as well. Mm-hmm. If you look at the Asian emoticons, most of what Rachel has shown as results here is directly reflected in them, including the confused expression.
Right.
Rachel, if I can just say, I really like how you don't impose anything on the animations, and so how natural do your subjects tell you those facial movements are?
That's a good point, actually. So all of the action unit combinations we use are biologically legitimate. Okay. Of course, if any of the facial movements don't look like something you know or looks weird, we say choose other. We don't really have many comments, people saying that looks really weird, or that they didn't like it. So they are only asked to choose a label if they think, yeah, that's definitely happy, or that's definitely confused.
So in your experience, they all seem to be biologically plausible.
They are all biologically plausible.
Okay.
Yeah. Yeah.
Philippe, can I ask you one question? So you started with a 15 millisecond difference between laterality, and then I think with your redundancy data, I think you answered my question, but I just want to know the answer to this. Is it side-based or is it information-based? That is, if you flip the face, would the brain know? And if you did a chimeric face and had like two different sides, like would it know? Does that make any sense at all?
Yes, it does. It certainly would know if the brain knew the identity, because it's something you can spot quite easily when people flip them, because they just look different.
Is it just doing side, or does it know the information on that side? That's really my question. Is the right side doing the left, or does it know what's over there?
Yeah, it's okay. So everything I've shown basically, and everything we've done with these experiments, we've pretty much stopped at 200 milliseconds.
Yeah.
So up to 200 milliseconds, the left and right brain seem to be operating pretty much in parallel, doing a coding of the face that can be task dependent. Now remember, when we do this reverse correlation analysis, at least in these designs, we could make them more sophisticated. We just group all the trials together. So for all I know what happens after 200 milliseconds, they could use the left part, they could use the right part equally. I don't know that yet. But it's something that we are planning to develop more, because with the sort of network that we reconstruct, there are some operations that we believe we can start to study, knowing roughly when they should happen and when they should not happen. So what's very interesting, and I did not elaborate on this because we haven't done much on this, is that you find in early visual cortex coding of features that are not task dependent. But then you see no trace of them subsequently when you look at the more occipitotemporal sources. However, you see convergence of features there. So this would be an ideal type of paradigm to study blocking of feature or lack of attention to feature or stoppage of feature versus feature integration for the purpose of a task. And this is when, of course, if you do not have information that must be integrated, that is where you must perform a logical AND on it, you can do it as an OR, which is at the moment what our analysis doesn't dissociate. So in the context of addressing really algorithmics, that is, are we observing at this node an OR or an AND, a formal operation, we need to change our design to something more subtle. Do I'll start from the left to the right.
A bit like Paul's question. You showed us how the information was transferred from the eye on the left in the contralateral hemisphere to the ipsilateral hemisphere, so somehow building up a bilateral representation. And I assume the other way as well.
Happens as well, yes.
But what would happen if the face was fixated off center, so that both eyes were in the left hemisphere?
That's precisely what we are doing now.
And
we don't have an answer yet. But yes, of course, that's the way to develop those designs in order to start to address those sorts of questions. I'm doing this with Guillaume Rousselet, by the way.
Yeah, I guess the question is whether the coordinates for the analysis are face-based or retinally based.
Yes. No, this I can address. They are phase-based, because what we've done already, without presenting them left or right, is change the scale. And then you can see that the N170 tracks the location of the eye. And we've done also this, I mean, there are studies done with bubbles by the Di Carlo group in monkeys where they look at the face patch and they controlled for scale, for translation, and they find indeed a response that's not invariant but robust to these changes. And we've done also this with body parts, with vogels, where we find the same properties with macaque monkeys as well.
So
Just go like this.
Rachel, you mentioned briefly that you are experimenting with changing morphology of faces to change how the actor, say, like, if the actor was a different ethnicity, like how that would affect it. So, and it seemed like most of your data was about the observer, how the culture of the observer dictates the perception of emotions, but does it also, so if the performer was of a different culture, how does that play a part into emotion perception?
Do you mean a different culture or ethnicity?
Or like,
It's different.
Explicit.
So do you mean maybe we change the stimulus? So let's imagine we have a Western facial expression and we display this on a East Asian identity versus a white Caucasian identity. Is that what you mean?
Or like, do Asians have different expectations for how a Westerner versus an Asian would display emotion?
Okay, so in the PNAS paper, we had Easterners and Westerners observers, and they viewed same and other race faces. Because we thought what we might find is that we might find there might be effect of the face, the race of the face of the stimulus. But we didn't really find anything like that. And that's why they did so many trials, because it was same and other race faces. But I'm very interested to try to combine the dynamics with different face morphologies and identities to see how these combine together. And we have a paper, a psych science paper in 2014 that shows that with face morphology that is judged as, say, very untrustworthy, if you then add a specific dynamic to that face, it can be judged as very trustworthy. So we actually show that dynamics overrides morphology within at least social traits.
Do you look at the eye movement at all when you're looking at how the observers process emotional faces?
No, because it's fixation free. So any of the action units that are correlated with response could be foveated or otherwise.
So following up on Paul and Patrick's question, what about coding of, for example, like the symmetry? So it probably involves more than one bubble, two or more bubbles.
Yes.
So is that any way to deal with So like, so let's say some facial expression will be asymmetric
Yes.
expression, like one eye is closed, another open, versus both eyes open widely.
Yeah, that's to be frank, we've never done that. We've not looked at asymmetric expression so far. We've used primarily with brain data static images of fax-coded facial expressions. We've not gone into asymmetric or chimeric faces as was suggested before, but I could definitely see the point of doing something like this.
Right, so because I think it's adding a component of relations between bubbles instead of just using different size bubbles. Sorry,
Yeah, if you want, I mean, you can build up the bubbles paradigm from univariate, as I've shown here with what we have, to multivariate. I mean, there's no problem there. You just build specific relationships within the bubbles, and you sample on the relationships themselves, rather than sample on the univariate locations. Yeah, it's a very good idea. It's something to be done. We haven't done it. We've moved quite quickly from there into the designs that are more tailored to the questions that interested us then, which were questions of facial movements and expressions. If we had been interested in issues of symmetry, such as those you mentioned, yes, sampling relationships would be a very good thing to do. So I'm going, yes?
So if I understand correctly, the features are the ones that are most diagnostic for the behavior performance, right?
Yes.
So can you, or have you looked at you're looking for how a specific feature moves across a time course?
Yes.
Is it possible to look to see across individuals whether the point at which, so that sort of like if there's no mutual information at a later time point for certain areas, for certain electrodes, is that diagnostic of whether, reaction time differences or whether people make errors in their performance? So if a single feature doesn't progress beyond a certain electrode for individuals, the information is there in the brain perhaps, but it's not reaching the location required to now translate into action.
Yeah. There's a massive elephant in the room, and it's called semantics. So at some point, feature coding translates into much more complicated semantic information, which of course, as of now, we don't have the means to track in the same way. There are ways of doing this, obviously, which would be to get independent ratings. Say, for example, facial expressions of emotion are great for this. You can get ratings along the typical dimensions that they use in this literature. And then you can regress them to see which regions get involved. And then, because these are essentially regressions as well, you can look at how this relates then to the features, whether you have net gains when they are known semantics versus not, and so on and so forth. I have a big grant that's planning to do a lot of this. It's starting soon, and there will soon be ads, so we'll be able if people are interested to join this project, they'll be welcome. But yeah, that's something that we are very, very keen to pursue. We have clear ideas of how to do it as well. But at the moment, we haven't. What I can tell you is that at the moment, there's an experiment that's being analyzed where subjects did a 6-choice task on expressions, on males and females. And then what we can do with all of the tools we have is look at task modulations of each brain source. We have a much better localization than in this particular network as well. We have very good localization. We've used a beamformer, not IC. So we can see which source is task modulated, which source is feature modulated, which source is modulated by diagnostic feature versus common feature, and we use the nice property of the additivity of mutual information to basically, and the conditioning to get rid of all sorts of variables that we don't want to see, to isolate the effect, say, of a task. And when we do this, we find that, as with the very first experiment I've shown with the P300 being mostly task sensitive, we find big peaks of task sensitivity on low parietal sources this time that also code mainly task dependent features. So it's, yeah, there's a lot of interesting stuff, but there's also a lot of stuff that need to be done to understand where semantic, where and when it kicks in.
We should stop the public questioning now so people can escape if they need to. But we and Rachel are here to answer some questions informally afterwards, and they'll be around all day tomorrow too. Thank you.
Thank you very much.
Thank you.